Hello and welcome back to this lecture series about synaptic transmission. My name is Ranjita, and we are asking ourselves how neurons communicate. As you might remember from the last lecture, one of the first steps in synaptic transmission is the synthesis of neurotransmitters. Neurotransmitters are chemical messengers which enable neurons to communicate with one another, as well as with muscles and glands. The precise regulation of this communication is crucial for physiological processes as well as psychological functions, such as the regulation of muscles, mood regulation, sleep-wake cycles, cognition, and many more. To date, more than 100 neurotransmitters have been identified. In this lecture, we are going to ask ourselves two questions. Firstly, what are the fundamental properties of neurotransmitters? And secondly, how do we monitor them? The hallmark discovery that neurotransmission can be chemical is attributed to two prominent neuroscientists, Sir Henry Dale and Professor Otto Loewi. While investigating the effects of certain compounds on the nervous system in 1914, Sir Henry Dale discovered that extracts from the ergot fungus can have similar effects to stimulating the parasympathetic nervous system, which is responsible for processes such as digestion and muscle relaxation. He discovered that the substance in this extract was acetylcholine. And he noticed that acetylcholine can slow down the muscle as well as relax the muscle and slow down the heart. He also discovered that acetylcholine can be broken down by an enzyme, and this explains why the effect is so short-lived. In 1921, Professor Otto Loewi strengthened Dale's hypothesis about the effects of acetylcholine. He showed that not only can neurotransmission take place in a chemical way, in an electrical way, but also in a chemical way. He used a frog heart connected to its vagus nerve. The vagus nerve is responsible for regulating the heartbeat. And when he stimulated the vagus nerve, the heart slowed down. Then he used a second frog heart. He took the fluid from the first frog heart and bathed the second frog heart in this fluid. And quite surprisingly, the second frog heart also slowed down. This was a major turning point in showing that neurotransmission can be chemical. It was later found out that the substance responsible for this mechanism was acetylcholine. And for their hallmark discovery that neurotransmission can be chemical, Sir Henry Dale and Professor Otto Loewi were awarded the Nobel Prize in Physiology or Medicine in 1936. In order to be qualified to become a neurotransmitter, a molecule has to fulfill 4 different criteria. The first criterion is that it has to be synthesized in the neuron. These are specialized systems where particular neurons synthesize particular neurotransmitters. For instance, cholinergic neurons synthesize acetylcholine. Secondly, it has to be present in the presynaptic terminal and be released at amounts sufficient to have an effect on the postsynaptic neuron. This involves being stored in vesicles and being released upon nerve stimulation. For instance, glutamate is stored in synaptic vesicles and exert excitatory effects on the postsynapse when it is released. Thirdly, when administered as a drug, it has to have the same effect as in the body. In the experiments by Sir Henry Dale that we saw earlier, acetylcholine fulfills this criterion. And finally, there has to be a mechanism which removes it from the synaptic cleft. And this is to terminate the signal. If this doesn't take place, the neuron can be overstimulated and even cellular damage can take place. There are two groups of neurotransmitters. The first group are small molecule neurotransmitters. These include acetylcholine, which is involved in learning and memory, in smooth muscle contraction, cardiac muscle relaxation, also glutamate, which is the most common neurotransmitter in the central nervous system and is involved in learning and memory. GABA, which is an inhibitory neurotransmitter and is responsible for keeping the balance in the brain. And also glycine, which is another inhibitory neurotransmitter, which is mostly found in the spinal cord. Dopamine is a neuromodulator, but it belongs to the group of small molecule neurotransmitters and is involved in reward and pleasure and motivation. The second group of neurotransmitters are neuropeptides. Neuropeptides include endorphins, which are natural painkillers, somatostatin, which is involved in cognitive processing, as well as substance P, which is involved in pain signaling. The differences between small molecule transmitters and neuropeptides mostly lie in synthesis, storage, and mechanisms of exocytosis. Small molecule neurotransmitters are synthesized in presynaptic terminals from precursor molecules, whereas neuropeptides are synthesized in the soma. Small molecule neurotransmitters are stored in synaptic vesicles, whereas neuropeptides are stored in large dense core vesicles. Synaptic vesicles have more proteins in their release machinery, which are linked to localized release and recycling. Large dense core vesicles are not specialized to neurons, and they don't necessarily release from active zones, such as in the case of synaptic vesicles. Large dense core vesicles need a general rise in calcium, as opposed to the local rise in calcium at the active zone for synaptic vesicles. So they need higher stimulation frequencies in order to be released. Also, large dense core vesicles are only released once. They're only used once, whereas synaptic vesicles are recycled by mechanisms like clathrin-mediated endocytosis, kiss and run endocytosis, and bulk endocytosis. It was postulated by Henry Dale that all the synapses of a neuron release the same neurotransmitter. In other words, one neuron, one neurotransmitter. But this notion has proved to be wrong. Small molecule transmitters can be co-released, such as in the case of dopamine and glutamate in the hippocampus, or GABA and acetylcholine in the cortex and the retina. In addition to this, also neuropeptides can be co-released with small molecule neurotransmitters to modulate their functions. For instance, substance P is co-released with glutamate in the spinal cord to modulate its function in pain signaling. Co-release is an emerging research area, and it adds many layers of complexity to neuronal communication. The question arises, how do we monitor neurotransmitters? An ideal method would be minimally invasive, large scale, allow long-term measurements, have a high molecular sensitivity as well as a high spatiotemporal resolution. Each method has its own advantages. But the method we choose depend on the scientific question we ask and the experimental requirements. One common method to monitor neurotransmission is patch clamp. In patch clamp, an electrode is placed in the presynaptic neuron and it stimulates it. And when the neurotransmitter has been released and has bound to the postsynaptic receptor, we can measure with another electrode in the postsynaptic neuron the change in membrane potential. But since we measure the membrane potential rather than the concentration change in the neurotransmitter, this is a rather indirect way of looking at neurotransmission or at the neurotransmitter as such. It has temporal resolution of sub-millisecond scale, but the spatial resolution is rather low, as we can mostly measure from soma, but not from individual synapses in most cases. It is also limited to in vitro measurement. Another common method is microdialysis. In microdialysis, a perfusate is pumped into the brain via a probe, as seen in this picture, and the dialysate with the neurotransmitters and the neuromodulators is collected. In electrochemical techniques, an electrode is implanted in the brain, and this electrode senses the changes in concentration of neurotransmitter depending on its oxidized or reduced states. In one of the methods, a voltage change is applied, and the resulting change in current is read by the electrode. In positron emission tomography, radioactive tracers are used to label neurotransmitter receptors and their transporters. And this can be visualized using a PET scanner, their distribution and activity. Magnetic resonance spectroscopy is another common method which is non-invasive, and we use it to measure with the help of MRI, magnetic resonance imaging. We used it to measure the concentration of neurotransmitters such as GABA and glutamate. The use of biosensors involve wearable or implanted devices, and these can measure the concentration of neurotransmitter in bodily fluids such as blood. They're very often dependent on enzymatic reactions, which produce detectable signals when they interact with neurotransmitters. High-performance liquid chromatography is a laboratory method used for detecting, quantifying, and separating neurotransmitters in the blood or in the urine or cerebrospinal fluid. In immunohistochemistry, fluorescent probes, fluorescent molecules are used, or antibodies are used to label neurotransmitters in tissue sample. This can be visualized in the microscope, and the fluorescent protein can also be genetically encoded and bound to the ligand binding domain of the receptor. And upon conformational change, when the receptor binds to the ligand, the fluorescent protein becomes active. Neurotransmitters can have three different types of effects: excitatory, inhibitory, or modulatory. Whether it is excitatory, inhibitory, or modulatory depends on the receptor that the neurotransmitter interacts with. For instance, acetylcholine, when it interacts with nicotinic receptors, it acts in an excitatory way, whereas when it interacts with muscarinic receptors, it acts in an inhibitory way. Glutamate receptors, when they interact with their ionotropic receptors, they're excitatory, whereas when they interact with their metabotropic receptors, they're modulatory. The excitatory effect makes the signal continue by increasing the likelihood that the postsynaptic neuron will fire an action potential. And examples of this are glutamate interacting with their ionotropic receptors. Inhibitory effects block the signal, and by decreasing the likelihood that the postsynaptic neuron will fire an action potential. And examples of this is GABA acting at their inotropic receptors. Modulatory effects tweak the effect of other transmitters and have a long-term action. And examples of this is dopamine, serotonin, norepinephrine acting at the G protein coupled receptors. So in this lecture, we have talked about the fundamental properties of neurotransmitters. We will delve more into depth and talk about small molecule neurotransmitters, their structures and the different types, as well as neuropeptides. I hope you have enjoyed listening. Thank you.